Today, let me talk about next generation DNA sequencing. This is the overview of my presentation. Begin with DNA sequencing and end with transsequencing. DNA sequencing is a laboratory technique used to determine the exact sequence of bases such as adenine, cytosine, guanine, and thymine in a DNA molecule. The DNA-based sequence carries the information of a cell needs to assemble the protein and the RNA molecule. DNA sequence information is important to scientists for investigating the function of genes. The technology of DNA sequencing was made faster and less expensive as a part of a human genome project. DNA consists of a linear string of nucleotides or bases. For simplicity, referred to by the first letter of their chemical name. Adenine for A for adenine, T for thymine, C for cytosine, and G for guanine. The process of deducing the order of the nucleotides in DNA is called DNA sequencing. Since the DNA sequence confers information that the cell used to make RNA molecule and protein, establishing the sequence of DNA is key for understanding how genomes work. The DNA sequencing is the process of determining the nucleic acid sequence, or the order of the nucleotides in DNA. This figure or diagram shows A, DNA to be sequenced. The analysis for the Sanger and Maxam-Gilbert method begins with isolating the DNA, then amplification to increase the amount of DNA you want to analyze. This amplified DNA will be used as a template. The B picture shows the Sanger method. The four types of BNTP and BDNTP were included in the sample. BDNTP was assigned a marker in the form of a compound that could fluorescence and radiate. Then polymerization is carried out with the help of DNA polymerase. When BNTP enters one part, the polymerization reaction will stop. With this process, many DNA strands are obtained from DNA template of various lengths. The C pictures show the Maxam-Gilbert method. This method was developed earlier than the Sanger method, but after the Sanger method existence, the Maxam-Gilbert method was soon abundant. In this method, the 5' of the template DNA is first radioactively labeled as phosphate 32. Then the DNA template will be cut into 5 types of chemical, namely dimethyl sulfate or G, acid A and G, hydrogen with high salt C, and hydrogen C and G. That way, the DNA template will be cut into various sizes. After the two methods in the previous slide have been carried out, then the DNA will be analyzed by electrophoresis method. With this method, the DNA will be separated based on its size. Electrophoresis is a general term that describes the migration and separation of charged particles or ions under the influence of an electric field. An electrophoretic system consists of two electrodes of opposite charge connected by a conducting medium called an electrolyte. The separation effect and the ionic particle result from difference in their velocity, which is the product of the particle mobility and the field strength. Advanced method of sequencing involve shotgun sequencing and de novo sequencing. De novo sequencing is used to determine the sequence of desired DNA with no known sequence. Shotgun method is designed to analyze the sequence longer than 1000 base pair, where the large genome broken down into random fragments. The main difference between basic and advanced sequencing is in the amplification and analysis process in the advanced sequencing. In the advanced sequencing, the amplification and the analysis can be carried out on many DNA template strains that you want to analyze. This can be done because the DNA strain is immobilized first in a medium. The A picture shows the immobilization process with microbit. One strain of template DNA is attached to one microbit. Then all of them are amplified simultaneously to produce microbits with many strains of the same template of DNA. The B picture shows the process of immobilization in planar media. The template DNA strains were attached to the planar media and then amplified by the bridge amplification method. In that way, on the planar medium, similar DNA cluster will be formed. Microbit that already have multiple DNA templates are separated into picotiters so that one picotiter has only one specific microbit. Then there are two ways that can be done, namely 405 for pyrosequencing and ion torrent. 405 for pyrosequencing is one type of fluorescence labeled DNTP that added to the sample each cycle. In each cycle, in its microbit, the fluorescence intensity was checked. Each bit is then placed in a single well of a slide. So, each well will contain a single bit covered in many PCR copies of a single sequence. The wells also contain DNA polymerase and sequencing buffer. The next method is torrent ion. The torrent ion is one type of DNTP that added to the sample every cycle. At each cycle in microbit, the pH change that occur are checked. Template DNA cluster formed on flat media. In each cycle, 4 types of fluorescence DNTP were added, and a blocker was added so that in each cycle, there was only one DNTP attached to the DNA template. In each cycle, there is a check for the fluorescence color form. This chapter will explain about the improvement, the basic concept, and the example of next generation sequencing. The four main advantages of next generation sequencing over classical Sanger sequencing are: the first is a sample size, and in next generation sequencing is significantly cheaper, quicker, needs significantly less DNA, and is more accurate and reliable than Sanger sequencing. Let us look at this more closely. For Sanger sequencing, a large amount of template DNA is needed for each read. Several strains of template DNA are needed for each base being sequenced. As a strain that terminates on its basis is needed to construct a full strain sequence. In next generation sequencing, a sequence can be obtained from a single strain. In both kinds of sequencing, multiple staggered copies are taken for continue the construction and the sequence validation. And then the next advantage is speed. Next generation sequencing is quicker than standard sequencing in two ways. Firstly, the chemical reaction may be combined with the signal detection in some version of next generation sequencing, whereas in standard sequencing, these are two separate processes. Secondly, and more significantly, only one read can be taken at a time in standard sequencing, whereas in next generation sequencing is massively parallel, allowing 300 gigabase of DNA to be read on a single run on a single chip. And then the next advantage is cost. The reduced time, manpower, and reagent in next generation sequencing mean that the costs are much lower. The first human genome sequence costs in region of 30 million pounds, but using modern standard sequencing method, aided by data from the known sequence, a full human genome would still cost 6 million pounds. Advantage is accuracy. Repeats are intrinsic to next generation sequencing, as each reach is amplified before sequencing and because it relies on many sort of overlapping reads. So each section of DNA or RNA is sequenced multiple times. Also, because it is so much quicker and cheaper, it is possible to do more repeat than with standard sequencing. More repeat means better coverage, which lead to a more accurate and reliable sequence, even if individual reads are less accurate from for images. This figure shows the basic concept of the third generation DNA sequencing nucleotide detection. The A figure shows the nucleotide detection in a zero-mode waveguide or GMW as pictured in biosequencer. In this method, DNA polymerase molecules are attached to the bottom of its GMW, and target DNA and fluorescence nucleotides are added. As the diameter is narrower than excitation light wavelength, illumination rapidly decay, traveling up the GMW nucleotides being incorporated during polymerization at the base of the GMW provide real-time bursts of fluorescent signal without undue interference from other label DNTP in solution. The B figure shows the nanopore DNA sequencing as employed in OMP-Minion sequencer. Double-stranded of DNA gets denatured by a processive enzyme, which tracks one of the strain through a biological nanopore, and then embedded in a synthetic membrane, across which a voltage is applied. As the single-strand DNA passes through the nanopore, the different bases prevent ionic flow in distinctive manner, allowing the sequence of the molecule to be interpreted by monitoring the current at each channel. This video shows the single molecule real-time or asymmetry with a zero mode waveguide. A single molecule real-time or asymmetry is a parallelized single molecule DNA sequencing method. This is utilized by a zero-mode waveguide or GMW. In this method, a single DNA polymerase enzyme is affixed at the bottom of GMW with a single molecule of DNA as a template. The GMW is a structure that creates an illuminate observation volume that is small enough to observe with only a single nucleotide of DNA being incorporated by DNA polymerase. And the next is how to nanopore sequencing analyze the DNA. The nanopore sequencing is a unique scalable technology that enables direct real-time analysis of long DNA or RNA fragments. It works by monitoring changes to an electrical current as nucleic acid are passed through a protein nanopore. The resulting signal is decoded to provide the specific DNA or RNA sequence. This video shows the nanopore sequencing reward.
Oxford Nanopore is developing a method of DNA analysis called strand sequencing. At the heart of strand sequencing is a protein nanopore. This model shows a typical nanopore made from protein. You can see that at the core of the protein is a hollow tube that is only a few nanometers in diameter. Oxford Nanopore designs and manufactures bespoke nanopore structures for a range of applications. In nature, nanopores form holes in membranes. In Oxford Nanopore's system, the nanopore is inserted into a membrane created by a synthetic polymer. This membrane has very high electronic resistance. Here you can see a nanopore piercing a single hole in a membrane made from synthetic polymer. A potential is applied across the membrane, resulting in a current flowing only through the aperture of the nanopore. Single molecules that enter the nanopore cause characteristic disruptions in the current. By measuring that disruption, the molecule can be identified. In strand sequencing, an intact DNA polymer is sequenced as it passes through the nanopore. Here you see a DNA enzyme complex approaching the nanopore shown in blue. The enzyme shown in green is designed to ratchet the DNA strand through the nanopore one base at a time. The enzyme binds to the end of a double strand of DNA and unzips the double strand to form a long single strand which it feeds through the nanopore. As the DNA strand moves through the nanopore one base at a time, a characteristic disruption in current is created by the presence of particular combinations of bases in a particular part of the nanopore. Because these disruptions in current are so specific to the different combinations, this information can be used to determine the order of bases on that DNA strand. There is no deterioration of accuracy as the long DNA strand is sequenced. By preparing the DNA so it has a hairpin structure at its end, the system can read both strands, that is, the sense and antisense strands of the DNA. This gives advantages in data analysis. In order to create a high-throughput system, a number of nanopore experiments can be conducted at the same time by using an array chip. Multiple microwells are fabricated onto an array chip using standard semiconductor materials. These array chips may be scaled according to need. The user may require tens of channels to hundreds of thousands of channels depending on the application. This array chip is built into a consumable cartridge, which also contains the fluidics required to run the chip. The analyte is added to the cartridge, which is then used in conjunction with an instrument, the gridiron node, for real-time data collection and analysis. The array chip is also designed to be used inside the miniaturized MinION instrument,